Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 79th seminar, and I call it a Columbia University Day. I came back from my 1-month trips to US after giving 9 talks, and I heard actually today's junior speaker also giving similar tour I did. And then I am now in Hawaii to give another seminar today. And it's my first time in Hawaii, and I have enjoyed my stay so far. I did so much thing, including meeting shark in the ocean, of course in the cage. And also I went to many places, the many movies actually filmed. Also the Roast, one of my favorite show, also filmed there. It was so fun to meet many friends last month, and our colleagues as well as young researchers. So thank all for the kind invitation and the financial support during that trips. I will continue my trips to Europe again after this Hawaii trip. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Harris Wang. He is a professor at Columbia University in New York City. Of course, he does not need any introduction because we all know him, but he is a pioneer with any technology development, including the famous MAZE, the genome engineering tool, and many microbiome engineering tools he developed in his lab for decades. He is one of the few geniuses whom I have met in my career, and it's my great honor to visit him and give a talk in 2021 when I did a seminar tour on East Coast. Harris, I truly thank you for your time and for your contribution to the scientific society. The virtual podium is all yours, and thank you again.
Thanks, Tae-sak, for that super generous and introduction. It's a really great pleasure to be here. I've known Tae-sak for over a decade and more, and it's really been great to be able to experience and share the growth of this community, and really want to really commend you for this forum in which to highlight kind of the young speakers and the rising stars in the field. It's so important to be able to foster the growth of this community here. So it's a really tremendous pleasure for me to be here today. I thought I'd spend my time just giving 3 quick slides on some aspects of our work and kind of my journey in science. If hopefully you guys can see this.
Yes.
Our lab is interested in developing kind of next generation technologies for both understanding and characterizing and manipulating the microbiome, as well as emerging cellular therapeutic platforms using systems synthetic biology approaches. We are particularly interested in this concept of microbiome gene therapy, where you can go in and really manipulate and program microbes that colonize the gastrointestinal tract so that they can have new functionalities like diagnosing a variety of diseases, as well as produce therapeutic functions. We're also really interested in application of microbes in other environments beyond just the human body. For example, in building materials or in soil and bioremediation. We're also excited about incorporating some of the super exciting advances in machine learning and genome engineering and design to create synthetic cells, as well as new CRISPR therapeutics for epigenetic kind of medicines. And so if you're interested in more on what we do, please go to our website or reach out to me about some of the things we're doing. I thought I kind of share with you my kind of journey, intellectual journey, and how I got to where I am today, and some of the key highlights that have shaped me as a scientist, and hopefully, sharing that story might be kind of interesting for folks. When I was growing up, since I was little, I was super excited about physics, right? So some of my, earliest memories about science was really thinking about really reading books about black holes and Albert Einstein and Stephen Hawking, and this was something that made me so excited about science. And so when I started undergraduate at MIT, I majored in science and in physics and mathematics, because I thought, well, those are pretty quantitative areas, and if I could kind of make it and try to at least get a major at MIT in physics, I will have a good solid quantitative background. And I would say that that's a generally important thing, is to be as rigorous as you can, as early as you can. Just because there's not as many opportunities to exercise that as you go on kind of in your career, just because you have so much, I think when you're young, you have so much more time to struggle with your problem sets, etc. But while I was at MIT, I actually explored a variety of different kind of areas of research, and I did research basically every semester. I worked on things like modeling cardiovascular disease, stroke and imaging kind of research, as well as learning and memory studies. And it was clear to me actually that I wasn't a very good physicist, right? Like I was not going to cut it in physics, but there was so much you can apply quantitative principles and rigor into biology, and that was something that really kind of propelled me. And so when I started graduate school at Harvard, I tried a few different things, and maybe this is one theme here, is to really immerse yourself in potentially very different topics, and maybe some things will kind of arise from that. Maybe not today, but maybe in 5 years, maybe in 10 years, or even longer. So I did stuff in drug delivery and kind of microfluidics and did rotations, but finally landed on a discussion with George Church. I was actually having I went to a talk, and there was an evening seminar, and I sat next to George, who is a pioneer in synthetic biology, and that really helped propel me to this next stage, where I was like, this is so exciting that we can go in and try to engineer biology in this very quantitatively rigorous way. And this was kind of at the early 2005, 2006 period, where synthetic biology was really starting to pick up in the United States, with the forming of SynBYSS, the NSF-funded synthetic biology engineering research centers, where Tae-sak and I met actually through a number of other investigators. It was led by Jay Kiesling at Berkeley, and so this really kind of to me was the start of this field, at least certainly for myself, where I was able to contribute to build some of the early genome engineering, genome editing technologies. And this is in bacteria. I learned a lot from both the molecular side, but also instrumentation and how do you scale this engineering to be able to produce biological systems in totally different ways. And then, I would say this is like the BC era, like before CRISPR era, right? And so, in 2012, 2013 was really kind of the usher, this new era of genome editing, where I got to see some of those really exciting advancements. And this is where I was at the Wyss Institute just after having graduated, and I really started to think about, well, how do I not only engineer a single cell, but maybe populations of cells? Because really in environments, it's really just populations of organisms that are out there. And so I started to really think about ways that we could do this at scale. And that's what I started to do when I really started at Columbia, was to really think about how microbes interact with one another. How can we understand their spatial temporal organization, as well as their metabolic interactions, and be able to build kind of next generation tools, be able to delineate them in time and space, and be able to engineer them with higher sophistication. And so kind of that's, brings me to my journey today of really trying to do that. So I guess I'll just really quickly give a few kind of advices to people. I thought I see personal advices for trainees that I thought was really important or useful for me. And one thing I learned early on was that it's always good to try to aim really high, right? Aim for the stars. Maybe you'll never get there, or you won't get there at least in the time frame that you thought. But along the way, being ambitious is very helpful because it sets you to potentially, even if you fail, to get to somewhere better than if you were to aim, not as high, right? And so I think being ambitiously driven in terms of setting your aims, being somewhat realistic, but also just, try to do the best you can, but, go as far as you can. Maybe you don't get to Mars, you get to the moon. That's still a great achievement. And then along with that is that throughout this process, one should always try to be productive in learning how to fail. And I think science is built on the failures of having tried something and not working. And if you can learn from that failure in a productive manner, then you can go very far, right? We didn't go to the moon by just, building one rocket ship and going up there. We tried many, many times. And it's the same thing with biology as it is in any other area. I think one other thing is really understanding incentive structures, right? Of not just kind of the organization you're in, the academic environment, but also your colleagues, right? Everyone is in a different part of their life in terms of where they are kind of in their career goals, and understanding what drives each of the individuals around you could help you to optimize how you can help them as well as yourself in that process, right? Sometimes I see a lot of clashes between individuals because they don't understand the other side, where people are trying to go. If you understand where they're trying to go in terms of their career goals, then you can maybe help with that, or at least understand where they're coming from and establishing good dialogue so that you can kind of minimize conflicts. I think it's super important to try to share as much as possible. Like, ideas are meant to be shared, and we need to share that as much as possible. Holding ideas close to your chest, I think, is always not a good idea, because it means that the idea has not been vetted by multiple people. And so, just like anything else, if you have really good ideas, it needs to be crafted over time and get iterated by many, many different individuals. And to me, that's something that I always try to do. It's so important to be kind. I think I don't need to overemphasize this. There's so many, really brilliant people everywhere, right? But it's so important to just be kind to everyone that you interact with, whether it's trainees, whether it's just someone who doesn't understand what you're doing or is just asking a question. Be generous about that. And I think that's something that we can all use more of. And so I would just suggest that everyone try to do this as much as possible. Stay intellectually curious and hungry. I think everyone knows this, but it's really true. The more you're hungry about learning, the more excited you get and the more you can sustain your science. And then also take breaks, right? Their life is a long journey. You have lots of things going on in your life, and make sure that you do this. And it's not a sprint. Sprinting to the next milestone doesn't mean that you'll be able to sustain for the next set of miles, right? And so really kind of do enjoy that with your friends, your family, or those that are close to you, and your colleagues. So that's all I had to say for this. So I'm gonna hand the podium back to Tesa.
Amazing, Harris. I always wonder why your path or career path similar to mine, because my career path started with the chemistry, your path started with the physics, and then now I'm, I did, metabolic engineering, and then now I'm also doing microbiome. And then now I found the reason, because you share your philosophy with me, similar one, and everything's wonderful. I mean, dream high, why not? And then at least you can come to me, the moon, and not Mars, and also you said learn from failure. amazing. That is the one thing I learned a lot from failure. So young people, I emphasize, don't worry about anything, just do it, fail it, and then you will learn more. And then be kind is one of the important things. And that's how Harris shared his very, very limited time today with us to give the wonderful, advice. Also, of course, take breaks. I probably take too many breaks right now, traveling too much. And that's amazing. I mean, sharing ideas with other people, that's amazing. So thank you so much. I mean, that's all wonderful advice for all the young people. Thank you.
Thank you.
Okay, so now the main speaker of today with a longer introduction, Professor Andras Gyorgy obtained his PhD from MIT. That's very interesting today. He and Harris and I actually have common thing. That's MIT today. So I need to call today MIT Day. And he got the PhD degree at MIT in 2016, working with Domitilla Del Vecchio, focusing on retroactivity and metabolic burden. And that is actually, his work is the pioneering work in the metabolic burden field. And after that, his paper came out, and basically everyone just cited that paper because that is the first paper, in the field. Following on 18-month postdoc with Murat, Arkat at UC Berkeley, he is now an assistant professor at New York University, Abu Dhabi. His research combines quantitative modeling and analysis with wet lab experiments targeting modularity, scalability, and context dependence in synthetic biology. Current active project center around dynamic plasmid copy number control, leveraging feedback architectures to accelerate and decelerate evolution, a very important topic, and designing and implementing genetic feedback optimizers. Interesting, as I said, we are all MIT alums. Also, Andras is the first speaker from Abu Dhabi or nearby regions, making this seminar truly a global event. Also, I heard today, Andras is quite a traveler. He grew up in Hungary and then did his PhD in United States. Now is in Abu Dhabi. And also he traveled just similar route recently, went to Switzerland to meet the Mustafa and then go to the Oxford and Imperial, and all three places was basically the place I went to for a seminar. So Andras, thanks so much for your time today, especially from the inconvenient time zone. And for me, it's also inconvenient time zone right now is 5:30. I'm so excited to listen to your talk, and please take it away. Thank you.
Thanks for the kind introduction, and thanks for organizing this. Let me share my screen, and let me quickly check if it looks good on your end as well, right?
Yeah, wonderful.
Okay,
go ahead.
Okay, so once again, thanks for organizing this, and thanks for the generous introduction. And today I would like to talk about one of our recent works, and the paper is titled exactly as the title of my talk. So it's going to be about plasmid copy number control in E. coli, and namely inducible plasmid copy number control. So I'm going to start with a brief overview of a couple of sample projects that we usually do in our group, and they usually combine biology and quantitative modeling. The degrees are changing across projects, but they usually involve both of these. So the one that I'm going to be talking about today is the first one that I would like to introduce. So we originally started about questions regarding metabolic burden and resource competition. And what we realized is it was fairly tedious to build one circuit and put it in a plasmid with a low copy version, and then do the experiment. And then if we didn't like it, or if we wanted to try it with a slightly higher copy number plasmid, we needed to start cloning again and transforming again. So it meant that this progress was very slow because we needed to do a bunch of cloning and transformation steps. So what we realized is we wanted to have a system where we can just put our circuit in a single plasmid that we can put in any strain of E. coli that we like, and just adding some external inducer molecule, just like we would do with transcriptional and translational regulation, we can regulate the plasmid copy number. So that's what we set out to do, because we realized that this tool was largely missing. So this is the talk that I'm going to be giving today, but I also wanted to talk about 2 other projects so that you have a better understanding of all the things that we are doing. So the second one is, let's say I'm given a pathway in a cell. What I'm showing you now is PPGPP mediated growth rate control. So this is PPGPP, this is reLA, and this is spot H. So the more cellular stress we have, reLA expression goes up, PPGPP goes up, ribosome production goes down, so growth rate goes down. What we can do is we can counteract it by expressing spot H. So for example, Domitilla Del Vecchio had a paper recently where externally they can induce spot H expression, regulate PPGPP so that they can counteract the effect of reLA. The problem is, as you start expressing spot H, you can overexpress it and put extra burden on the cells. So there's this nice medium spot that depends on all kinds of environmental conditions of spot H expression, which would give you maximal growth rate. The problem with this is you need to do external control of spot H. So what we asked is, could we do something where we measure the variable that we would like to control, let's say growth rate? We have a genetically encoded optimizer, and then we regulate spot H expression so that we automatically find the optimal concentration of PPGPP. And the idea here is that this optimizer module is a pathway agnostic optimizer module. So we didn't build it, we didn't design it for this pathway, but instead what we set out to do is to design an optimizer module that you can realize using common synthetic biology modules, and you can just put it on top of any pathway of your choosing. And the last thing that I would like to talk about just briefly as an introduction is we have constitutive activation, constitutive repression, activation via a self-activated regulator, or repression via a self-repressed regulator. And all these motifs are very common in regulatory networks. And functionally, they are doing exactly the same thing. So if there is an inducer signal, then in the presence of this inducer signal, the product is being produced. And in the absence of this inducer signal, the product is not being produced. So these are functionally equivalent regulatory schemes. So the question is, why does evolution choose this over that in a certain condition, or this over that in a certain condition, depending on environmental fluctuations, the time scale of mutations, and other factors? So this is the third topic that we are currently looking at. So these 3 should give a fairly good overview of the kinds of things that we want to do and we do in the lab. But today I'm only going to be talking about the first one. So the talk is structured as follows. I'm going to briefly introduce plasmid copy number control, and PCN from now on refers to plasmid copy number. Then I'm going to briefly explain how we implemented our plasmid, which is called TULIP, for tunable ligand inducible plasmid. Then I'm going to tell you a couple of things about its properties. And finally, I'm going to give a couple of application examples, how you could use our plasmid system. So as a motivation of why we wanted to have this tool is if we think about transcriptional and translational control, it's a fairly flexible and easy to do regulatory modality. We can talk about transcriptional factors, we can talk about stars, or we can talk about toe-hold switches. It's fairly easy to regulate with a small molecule inducer, transcriptional and translational activity. Compared with this, plasmid copy number control is much more inflexible. So typically, the most frequent application would be you pick a plasmid with a copy number
Oh, we lost him. Andreas?
to either a specific set of strains. Okay, good. Welcome back, everyone. So you're typically restricted to a specific set of strains or a specific set of plasmids. And instead of this, what we would like to do is truly a one-stop solution where we have a single plasmid with all the regulatory components it requires. We would like to have an inducible version of this where we add an inducer into the growth media, and we can just upregulate and downregulate dynamically in the middle of the experiment the plasmid copy number from low to high to medium, and put it in commonly used E. coli strains. So we would like to have as much flexibility and versatility as possible. So this is what we set out to do, and this is what I'm going to be talking about today. So how we built it is our next step. So once again, the name of the plasmid is TULIP for tunable ligand-inducible plasmid. So we started with the PSC101 origin of replication because it belongs to incompatibility group C. So if we can build this, it is going to work well with other plasmids as well. So just a quick recap of how this plasmid works. What we have is repA as a monomer is upregulating plasmid copy number, but it also has the potential to dimerize, and as a dimer, it represses its own expression. So that means that we have a positive feedback loop, and we also have a negative feedback loop for plasmid copy number control. So the balance of this is going to give us the plasmid copy number that we are going to end up with. And one easy way to regulate it is to modulate this dimerization dissociation constant. So if we make it weaker, that means that we are going to have if we make it weaker, that means that we are going to have only this positive feedback loop. If we make it stronger, we are going to have this negative feedback loop with varying strength. So what people did is they built a bunch of variants of repA with different dimerization strengths. And that means that if we pick repA version 0 versus repA version 7, we can measure the plasmid copy number, the fixed plasmid copy number for every variant. So for every variant of repA, you're going to end up with one static copy number. And if we put GFP expression on this plasmid, what we can do is we can measure fluorescence expression of GFP, and we can measure the plasmid copy number via qPCR. And what we see is a nice linear correlation between the two, which means that from now on, we can use just a simple fluorescence measurement as a proxy for plasmid copy number control. So this is our initial point for our work. So what we wanted to do is build first a two-plasmid system for copy number control as an intermediate step. So what we did, we chose the repA variant with tight onto inhibition. So there's a low basal expression of repA. And from a second plasmid, we can add inducible repA expression. So this is now a two-plasmid implementation, where with this helper plasmid, with the induction of cuminic acid, we are producing more repA, and we can upregulate the copy number of this second plasmid. And the more cuminic acid we add, the more GFP expression we get, and the more GFP expression we get, that means that the higher copy number this plasmid is. So this is now a two-plasmid So with this implementation, what we have is, as we start adding cumic acid to the growth media, we can upregulate repA expression, and that means we can upregulate plasmid copy number. So that means that more cumic acid, more repA, more. So we modified these two RBSs here, and we screened for a variants that have a nice dynamic range with cumic acid induction, and that grew fairly well comparable to the wild type. And with that, what we ended up with is, as we start adding So as we start adding more cumic acid, which means that as we go from left to right, we start increasing plasmid copy number, the growth rate doesn't really change at all. So that means that we can really add cumic acid, upregulate and downregulate copy number, and cells grow pretty much the same way. So this is how we implemented TULIP, or single-plasmid tunable system. So next, what I would like to talk about is most important properties of TULIP, which is portability across strains and different media. How can we dynamically regulate copy number in the middle of the experiment, and its stability over time. So first of all, we wanted to check if TULIP is indeed working across all commonly used E. coli strains. So we selected the ones that we typically use in the lab. And although quantitatively, the behavior is different. So depending on the strain, the maximum copy number that we can achieve can vary from 40 to about 200. The qualitative behavior is the same without cumic acid. We have a low copy version, and as we start increasing cumic acid, plasmid copy number increases. So that means that our system TULIP is working as intended across commonly used E. coli strains. As we add cumic acid to the growth media, plasmid copy number goes up with a nice dynamic range that, of course, depends on what strains you're using. Of course, what we wanted to also test is, does it work in all kinds of medias that one would typically use in experiments? So we tested M9 with glucose, glycerol, LB media, and SOB media. Darker shades correspond to higher cumic acid concentrations, so higher plasmid copy number supposedly. And what we see is, again, the numbers that we have across media might change. So quantitatively, the behavior is different, but qualitatively, it's pretty much the same. As we start increasing cumic acid concentration, so we go from lighter to darker shades, plasmid copy number indeed goes up. And again, what we have is in this plot is across all media conditions, as we start increasing plasmid copy number, the growth rate doesn't really go down. So it seems that even though we are upregulating plasmid copy number fairly substantially, cells don't really sense any stress at all. So at this point, what we have is our single plasmid system works across different strains and different media that we usually use in the lab. So next, what we wanted to see is, can we really upregulate and downregulate plasmid copy number in the middle of the experiment, and how long does it take? And once we settle to a new set point, does copy number really stay there? So what we did is we set up a couple of KaiBios, and we used them in turbidostat mode. So every 12 hours, we changed the fresh media that we are pumping in, and the fresh media contains different concentrations of cumic acid. So what we have is we have a low concentration of cumic acid, so that should correspond to a low copy number. And we have a medium copy number, a high copy number, and a low to medium copy number. So what we should ideally see is, as we are looking at plasmid copy number via the proxy of green fluorescence expression, is we should start low, we should go higher, we should really go high at the 5 micromolar Cuma expression, and then we should fall back between the first two stages. And luckily, that's what we are seeing. So for the first 12 hours, we are in the low plasmid copy number mode. And then after a couple of hours, we settle into the medium copy. We further go up to the high copy. And finally, as we downregulate, we end up between the low and the medium copy number set point. And importantly, once again, the growth rate doesn't really change at all throughout these transitions. And what we also did is at the end of each phase, so just before transitioning to a new set point, we took flow cytometer samples to see the distribution. So gray shows you the distribution of non-fluorescent cells, and then this corresponds to the first set point change. So after 12 hours, this is the low copy mode, this is the medium copy mode, this is the high copy mode, and this is between the low and the medium copy modes. So the important takeaway from here is we have nice tight unimodal distributions after 12 hours in the new media. So for all intents and purposes, it seems that plasmid copy number is tightly regulated. And we also ran these experiments for 48 hours plus, I think, and with using just one cuminic acid concentration, and the lines are completely flat, which means that for extended periods of time, we can maintain plasmid copy number with similar distributions, just like we would get with traditional plasmid with static copy numbers. So next, what we wanted to test is with higher resolution in antibiotic selection pressure. Do we maintain the copy number every 2 hours? We took flow cytometer samples, and these are the medians that you see. And independent of the copy number that we set, we see that it is maintained as time goes by. So we are not losing or gaining any copies over time. And again, more importantly, what we have is without antibiotic selection pressure, we are losing the plasmid as we should. So if your application examples, you would like to have the plasmid for some time and then gradually lose it, just by changing the growth media, by removing cuminic acid, you can just lose TULIP from your system. So what we have now is, regarding the basic properties of TULIP, is it's a single plasmid implementation. It works across different strains of E. coli. It works across different growth media. We can really regulate it dynamically in a couple of hours. So as we are changing the media in 3-4 hours, the plasmid copy number settles to a new set point, and then it's stably maintained. If you have antibiotic selection pressure, and if you remove the selection pressure, you can lose the plasmid if you choose to do so. So these are the basic properties of TULIP, and then I would like to move on to the application examples. So first of all, do circuits behave as we expect them to in TULIP? Can we tune them? Can we prototype circuits? Can we use TULIP and regular plasmids in an interchangeable fashion? And can we acid? So first of all, we tested genetic circuits that are simple sensors. So this is an AHL inducible scarlet sensor, and this is a vanillic acid inducible scarlet sensor. And we tested them, and of course, the more AHL, the more scarlet expression, the more vanillic acid, the more scarlet expression. So everything works as it should, and darker shades correspond to higher copy numbers because we increase cuminic acid. So first of all, instead of building 3 different versions of this circuit using slightly different plasmid with fixed copy numbers, we have to just do 1 cloning and 1 transformation, and then change the cuminic acid concentration in the media to get different responses to AHL. So we can go from here to here without any cloning or transformation. Second, what we also tested is, this is now red expression versus green expression. So this is inducible scarlet, and this is constitutive GFP. And we previously had some work on metabolic burden, and what we saw there using plasmid with fixed copy number, with fixed static copy numbers, is in such a scenario, as we start expressing more and more scarlet, GFP expression should go down because of metabolic burden and competition for shared resources. And more importantly, it should go down in a linear fashion. So as we move to more scarlet expression, GFP should fall along linear lines. And what we have, again, across different plasmid copy numbers in both implementations is this seemed to hold with TULIP as well. So the take-home message from this slide is simple circuits behave as expected, and we can change their behavior easily by changing cuminic acid without doing any more cloning or transformation. So using the same two circuits, what we built is two different variants where we changed the RBS from weak to strong for both of these, for Luxar and Van Ram. So what we have first is we tested it in DH10B, both of them separately, and dashed corresponds to no induction, and solid lines correspond to AHL and then Van induction, and darker shade corresponds to stronger RBS. And let's say that our design specification is in the on state, in the induced state, we would like to have at least this much scarlet expression. So anything that gives us higher scarlet expression is a good design, and anything below is a bad design. So as we change plasmid copy number for the first circuit, we see that for the stronger RBS, the darker red, we have a fairly substantial range where we meet this design specification, whereas for the weaker RBS, we have fairly small range where we get enough scarlet expression. This is exactly the other way around for the very similar other circuit that we built. For example, what we have is we need to be in this very, very tight and narrow range for the stronger RBS, whereas for the weaker RBS, we have considerable leeway. And similarly, what we can see is for the first circuit that we have here, as we start increasing plasmid copy number in the off state, so the dashed lines, scarlet expression goes up. That means that we shouldn't have any scarlet to begin with. So that means that as we start increasing plasmid copy number, this promoter must be leaky. Compared with the other promoter in the other circuit that stays fairly flat as we start increasing plasmid copy number. So we can immediately conclude that while this promoter is not leaky, this promoter is fairly leaky. So again, we didn't do any cloning and transformation steps. We just changed plasmid copy number by a change in growth media, and we can easily see which promoter is leaky and which promoter is not. So what we can also do is we can now move into a slightly different strain, so from DH10B to BW, and we can reconfigure plasmid copy number and see if we can match the same design specification. And for the same circuit, we see a very similar behavior. So it's easy to change the plasmid copy number so that it works both with B0034 and B0032. But with the other circuit, what we have is for the stronger RBS variant, there's just nothing we can do in BW because there is no copy number that would give us the desired behavior. So that means that we can do a bunch of cloning and transformation steps and sample all this plasmid copy number range using all kinds of plasmids, but it would just not work, and it would take a lot of time to realize that. Contrasting that with using TULIP, we just do a couple experiments by changing the growth media, and that's the end of it. So finally, we wanted to do the testing of TULIP with a more complicated circuit. And also we wanted to really tease out if TULIP can work in place of regular plasmids, meaning can we prototype and tune circuits in TULIP and then put them in regular plasmids with fixed static copy number and expect that they behave similarly. So we started working with a toggle switch, and this is a standard toggle switch. So one side is red, the other is green, and with IPTG and ATC, we can tilt the toggle either red or green. And the experimental timeline that we followed is the following. So first we set the copy number by cuminic acid induction, and then we tilt the toggle using IPTG to green or ATC to the red state. Then we wait, we take the pre-washed samples. These are going to be the circles. Then we remove the inducers. So at this point, we are testing if the toggle remains in the red or in the green state. We grow them again, and we take the post-wash samples, and those would be the triangles. And this is flow cytometer data for the following slides. So first, we put the toggle switch into TULIP, and we changed the copy number by using cuminic acid, and we measured both green and red expression. So circles are pre-wash samples, triangles are post-wash samples. Red corresponds to the toggle that was induced to be in the red state. Green corresponds to the toggle that was induced to the green state. And darker shades correspond to higher concentration of cuminic acid, so higher copy number. And what we can see is whatever was induced green became green and it stayed green. Whatever was induced red became red and it stayed red. The only slight change from this pattern is the lowest copy, so the lightest green, because in the pre-wash sample, this is the circle. This is how much green we are getting. And in the post-wash sample, we are getting basically no green at all. So this toggle is pretty much losing the green state. So what we did next is we put the same toggle into plasmid with fixed copy numbers. So now every single implementation is a slightly different plasmid with a slightly different copy number. And in the background, what we have is this data repeated. So the data with the fixed copy plasmid versions follow very closely what we have, at least qualitatively, the data that we got using TULIP with cuminic acid induction. So that's a good sign because the toggle behaves fairly similarly in both these implementations. So it would suggest that there is a way to prototype plasmids in TULIP and then move them to matching fixed copy vectors. So what we did is we wanted to really tease out how we can do this quantitatively. So we measured the GFP expression from the backbone, so that's constitutive GFP expression, so that we can match some cuminic acid concentration to a particular fixed copy plasmid, so that we can say that if we are seeing this much GFP from TULIP and we are seeing the same GFP from a fixed copy plasmid, then the copy numbers are matching, so that we have a comparable realization. And with that, what we did is this is now the constitutive GFP expression from the backbone across all figures. So this is a proxy for plasmid copy number, either using the fixed copy variants or TULIP. And this is now the GFP expression, a different GFP expression from the toggle plus the red expression from the toggle. Circles, again, correspond to the pre-wash samples. Triangles correspond to the post-wash samples. Red and green correspond to whether we tilted the toggle into the red or the green state. And now lighter shade corresponds to TULIP, so that's tunable plasmid, and darker shades correspond to fixed copy plasmids. And the important takeaway message from here is that lighter data points and darker data points across all plots are aligning fairly well, which means that once we control for plasmid copy number, we can go from TULIP to a fixed copy plasmid, and the behavior is going to be quantitatively very, very similar. And what we also saw is that this is the green point that I pointed out on the previous plot, where we are losing the green induction level. And we realized that we were working with a LACI plus strain, which would put extra pressure on decreasing GFP expression. So what we wanted to test is if we work with the LACI minus strain, so same strain, just LACI knocked out, that green should go slightly higher to improve the toggle's behavior. And that's exactly what we have. So we repeated the whole experiment. Again, lighter shaded symbols and darker shaded symbols are lining up, and that green just went up as we expected. So that means that behavior in TULIP and behavior in plasmid with fixed copy number is fairly similar. So again, we can move from one to the other. And finally, what we wanted to test is So far, we built a plasmid system with an inducible behavior, but it responds to cuminic acid, which is good because cuminic acid is something that we don't routinely use, such as like ATC. But what if you would like to have something that responds to not cuminic acid? So what people could do is follow the same steps that we did and build a slightly different variant where this is going to now respond to something else. So swap cuminic acid with whatever inducer you would like to have. The problem with that is it takes a fairly substantial amount of time. So instead of this, what we can do is we can keep TULIP as is, and we can add another layer of control on top of it. So in this case, we chose a CRISPR-AI-based additional layer of control, where we have an additional knob that we can tune. And for example, in this case, we can add more vanillic acid. We can start expressing more guide RNA. It comes together with DCAS9 and it represses syndrome expression. So this is another way of interacting with TULIP and regulate plasmid copy number. And the good thing about this is if we were to use this, swapping out the left-hand side of this is fairly easy. It doesn't require really any tuning, and you can use whatever inducer you would like to have to start expressing this guide RNA and interact with TULIP. So what we did is we built two variants of this, one that responds to vanillic acid and the other that responds to AHL, same guide RNA. And this way, this is now vanillic acid concentration, and this is AHL concentration. Cuminic acid concentration is not changing at all. And this is the dynamic range that we got, the gray region that we got with changing cuminic acid. And with just changing vanillic acid, again, cuminic acid is not changing. This is the behavior that we are getting for the Van inducible circuit and for the AHL inducible circuit. So we get pretty much the same dynamic range. Again, we are using the same implementation of TULIP with the carefully balanced feedback loops, and we are just interacting with it with an additional layer of control. So that means that if you would like to swap out vanillic acid to AHL or pH or temperature or something else altogether, you can easily do just by modifying this part. And with this, I would like to wrap up my presentation. So today I talked about inducible plasmid copy number control that works across commonly used E. coli strains, across media. We can dynamically regulate it mid-experiment. We can go up in copy number. We can go down in copy number. It is stably maintained. Circuits behave as expected. We can tune their behavior easily. We can see if a promoter is leaky or non-leaky. We can go between strains and modify the behavior of circuits just by changing the copy number. And we can use TULIP as a flexible backbone to find the copy number that we like, and then we can move on to the plasmid of choice that we would like to eventually maybe deploy our circuits in. And these two projects are other representative samples. This one just came out today, and this one is still currently under development. And with this, I would like to say a huge thanks of my team. So the work that I presented today is mainly the work of Shiv and Chentao, who has been wonderful throughout the whole process. And I cannot say a big enough thanks to them. And with this, again, thanks for organizing this awesome opportunity, and happy to take questions. Thank you.
Amazing talk. And also I want to apologize. I thought I lost you, Andras, but I guess my hotel internet is not stable. So I probably coming back, go in and out, right?
No worries about that.
Yeah. So, but you didn't have any problem in terms of keep speaking, right?
No. I mean, I stopped because the recording stopped, and I restarted when the recording restarted.
Oh, I see. Oh, that has some problems. I need to go back and then to see what's happened. Oh, by the way, thank you. That's one amazing talk. Let me see. Do I have any question? Okay. So I'll start. This is amazing, and then congratulations, your recent paper again. I think the Nature Communication, I guess.
Thank you.
My question is, I love your use of CRISPR-AI. It makes your approach modular, or your circuit modular and generalizable. because you can use any sensor in the
Yep.
future. One caveat would be adding additional genetic circuit module, including CRISPR-I. CRISPR is very big, could potentially give some additional burden that you probably
Sure.
want to also avoid. I mean, could you kind of comment on that part and how you're gonna address that problem?
So that's absolutely true. I think that what you're meaning is because of DCAS9 competition for DCAS9.
Right. So basically adding additional circuit that is module, that's a DCAS9 module to control the entire thing, giving additional burden, rather than, there are two things. One thing, you could change the sensor without adding Cas9, the Cas9 part. That is the simpler, but you need to develop everything from the scratch. And then that is not ideal, but by doing your way, it's easier and modular because you don't need to touch your original circuit, but that will give more resource demand. And that is my question.
So absolutely. What you have is if your circuit of interest is also working with DCAS9, then obviously there might be competition issues, crosstalk issues, and all kinds of those problems. However, if you don't want to do DCAS9, then this is a very easy way to add an additional layer of control.
Mhm.
But if you run into issues like that, then obviously what you can do is follow the steps what we did. And the second time is probably easier than the first time, and develop a TULIP that is primarily responding to cuminic acid, primarily responding to your inducer of choice and not cuminic acid. It should take some time, but again, once you build that, you have that and everybody can use it. And probably we don't want to have 20 different ones, but a couple that are useful.
Wonderful. Now I see the time is a little bit over the 11 a.m. Central US time. And let me check, do they have the question already put, but because of my out, they lost. I don't know. Anyway, I will close and then we will chat again as a offline thing. So thank all for joining and staying today. Again, I apologize for my unstable internet in this room, in a hotel room. We will meet again on May 11, Thursday, the same time, the same Zoom link. We'll have Professor Drew Andy from Stanford University and Professor Natalie Farney from Worcester Polytechnic Institute. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. Thanks. I'll stop recording. Just 1 second.